Okay, so on Tuesday, we talked about one particular classification algorithm called the CVM, SVM, support vector machine. Then we discussed the cell-cell communication analysis. So this computer session, we will first by looking at SVM implementation in MATLAB. The MATLAB, there's a function called fit SVM. This is a function to let you to use command line to do this SVM classification. We will visit this page. It's provided by documentation of MATLAB toolbox itself. We will look at this section to plot the decision boundary and margin lines for two classes of this SVM classifiers. Example. Then from the user perspective, if you don't want to use command line, you have this classification learner application. Remember last time we tried using this learner application to do neural network classification. So there's interface. There are other options, including SVM and a kernel-based SVM. So we will look at this using one of the example provided by MATLAB itself. So everything is inside the MATLAB environment. Then after that, we are going to jump out. We also have a small examples to show you the kernel function using this visualization method. Then we will jump out to see, okay, how can we install single cell 24XCT in order to use this method to predict cell-cell communications. Basically, the output will be a list of ligand receptor pairs which detect from the data you gave. And those ranked on the top, those are the lists that potentially to be detected in the data you have. So the data you will give include different cell types. You have to select which cell type and which is donor and which cell type is receiver. But this package is developed in Python, so we will take this chance to install the package in Python and then set up the running environment, allow MATLAB to communicate with Python to use the interface to do this analysis. So you don't have to learn Python or try Python command line yourself. Once you set it up, you should be able to run this analysis using the interface we provided. Then we will try to look at this example data and look the two genes I provided as a pair of examples of ligand receptors in this data set. You will be using Toolbox to do this by yourself and to download the data, process it, annotate automatically, and then to show the expression of these two genes. Maybe we can use this example and try 24XCT. You run after you set up the 24XCT, you run on this example data, see whether you can detect other ligand receptor genes. If we still have time, I will show you how to merge two data sets together using different examples. I showed some of this before, and we can do, if you have a processed data you saved in the hard drive as two separate files, you can combine them. You can also directly download two files without even saving it, just process in the memory directly. Then at the end, I will show you if you have customized marker gene database, how can you use that information to annotate the cell type? Because currently the cell type was annotated using PanGLODB database, which has been built in as a resource provided by the interface. But if you see, okay, the cell type I'm interested in are not in the database, I have my own list of genes. I have a gene names, and for the cell type have highly expressed of this gene, I will assign a name as this particular given cell type. I will show you how you can do it. All right. So let's first visit this page called FIT CS SVM. So to do it in the MATLAB. Just before that, let's do a version. Just let me know what version MATLAB you are using. So my version is, so this is related to the setup for the Python environment later, because we need to have to find the compatible Python version which comparable with this. So my version is R 2023B. So what's your MATLAB version on your machine? Same? Okay. Same one? Okay. So we needed to find the Python version. Could be 3.11, because current version is 3.12, which is the latest version. It's just updated last week. But maybe we need an older version in order to make this comparable with, we will check their table. Just make sure they can talk to each other. Okay, this is older version, so we need a relatively older version of that. But we don't have to worry about it right now. Now we do doc feed C SVM. So look at this documentation of SVM machinery function. Question? Mm-hmm. 2021, you will find the Python version which is compatible with it. Yeah, okay, yeah. So we do have machine 2021B. What other version you have? You have 2023B? 2021? Mm-hmm. Okay, so we have several different. It's okay, yeah. So Python has been around for a long time. So we have 2021. Is 2021A? B, okay. We also have 24, that's your A or B? B, this is the latest version. So, yeah, so let's see what version we need to have a Python in this different scenario. That we decide later. Okay, if you look at this function, it's a classification algorithm. So you need provide variables, okay, in a table. So this is a table, and then you say, okay, I want to have this data, have those numbers of variable to describe these data points, and I want to classify these data points into classes. So this response variable name is one of the variable inside the table to indicate the class of these different data points. Or you can use a matrix X, and the Y is the label of the classification. Okay. The variable, this input variables, there are no difference from other classification functions, they all uniform forms of these inputs. So let's move down and we will, so those are examples. If you want to try, you can have X and Y. Okay, Y is a species name. X is a description of this fish. There are different measurements. We will move down, not using this example, but using this here. Okay, I think that's the same example. We can copy the code here to get the data. So right-click, copy the code here, and paste into So now we have a measurement of fish with 100 fish and 2 variables. Okay, this could be the body length of the angle of the fin, whatever. There are 2 variables to describe 100 different fishes. And those fish are classified into 2 different species. That's S, and one is called Setosa, and then Virginica, 2 subspecies maybe. So that's our input. We need training data and the responder variables, which the class of the data points. So this is a classification problem for classified 100 different fish using 2 variables to train the model. So now we're going to do the training, and we don't have a new fish to come in to do for the prediction purpose, but we just train. This is a known data set. So we have training data. To do the training, it's very easy. You just put X and S in. Okay. Okay, inside, you know what's going on. There will be a hypothetical separation line between these 2 group of data points, and a lot of computation compute the data points to this line, the distance. Okay, all this computation was done using matrix multiplication. After that, then change and found, okay, the best solution at the end. So that's what's going on under this algorithm. Now, what we want to do is to look at, get some information out from this trained model. If you look at if you copy this, you get the value of beta and the B. So this is a Bayes term, which is the intercept, and this is beta is basically this is a slope, the coefficient of this linear prediction. And SV contains the data point with the support, the separation of the 2 classes. Those are the points which close to the line and in the boundary of this separation. Okay, these are called boundaries. So this SVM machine found those points and focus on those to build this prediction model, rather than to simulate the whole thing as a distribution. So it's a clear cut. They just found those most important points to separate them. So what we want to see is to plot XY out first. So this show you the fish, okay? So So this is called a scatter plot with a group. So you remember we have two variables in X, two separate fish, and then the fish is labeled with S. So this will plot as a, if you do the first two lines, this will give you the fish distribution. So group scatter will use the group variable which provided by S to separate those, to color code the different classes. Okay, so this is 100 points here. Now we copy the third line from the example. So this will plot a circle with black color around those points that have been found as a support vector points. So this line is just a highlight those points which has been selected by the SVM algorithm as a boundary points separate these two species. So this is a trained model, so beta and B define the separation, the straight line to separate. So we can plot this line using the information provided by the beta and the B. So that's where the straight line's definition is here. So you see the content slope information and also the bias. So we can copy this and plot onto the same figure by just do this. Okay, so this is the separation for the two species. There's one fish that's been put in this class, which is a classification. But I think this is the best solution and the most robust separation, because we want to keep the boundary margin as much as possible and tolerant one mistake given this point. Otherwise, this straight line maybe have to go through the very narrow region which is in between of this, right? So this algorithm using the soft cutoff. We can also plot out the margin. So you keep this, copy, and plot them. So this dashed line to show you the margin, upper and the low boundary. This boundary line located in the middle. Okay. So that's what we got from here. So after training, you can use the model. Let's see, the model we built is SV. So so this is data structure contains the separation line and also the margin region. So it's ready to take. I believe you can do the prediction to predict if you have a data, say X, the first fish. I'm just trying to see whether this is correct. So if I gave the first fish, remember the fish, you have 100 fish. If you give the first fish with 2 numbers, it will give you the name of species of the first fish. So if I have a fish, for example, if I have a point right here, this 7 and 2, it should be this Virginica. So if I put 2 numbers, 7 and 2, so this should give me Virginica if you look at this model, the training, to do the prediction. 7 is here and the 2 is here. So if I have a point located in the place where this yellow cursor is point 2, it should give you the Virginica. It's the right fish. Any questions for this part? Now we can look at how to use the classification learner to do this. Have this in your history of your command line. So if you type class and use them tab, you should be able very quickly to get this classification learner. Remember, L is capitalized, so the big L. Take some time to prepare this interface. If you have this section, this window ready, and we can start add a new session by click this. We need to load the input data. So input data contains select input is inside the big X, and responders also from workspace should be S. Okay, so this is our training data. Do you have the same thing? Let's see. New session, yeah. Just wait, you should have S in there. X, big X. Mm-hmm. And from a workspace. Okay. Yeah. Okay. Yeah, new session to allow you to start to put data in. Yeah. You should still have the variable x in there. Yes, the same problem. So we are just doing the same thing, just like you use a command line to do, but it's just provide an interface for you to. The range, no, now you select s as your variable. Oh, you have a y.
Saved as y.
Okay. Yeah. Yeah.
So should I start?
You should be okay. Yeah. 100 cells. Mm-hmm. Okay. That's no problem. That's right. Mm-hmm. Got it? Yes. Okay, good. So those algorithms have been so well developed. So it's a solve the problem. Basically, you require how you are going to creatively using them is a key. So if you pay attention, we have a homework. This homework is to allow you to spend some time to read that paper. That paper is one of the examples to using this like classification algorithm have been well developed, but used to a new question. So you just change the angle to see, okay, I can do classification to classify cell types, for example. But they use the same algorithm that's just defined, say, okay, which cell type I should use to separate patient from healthy individuals. Then they work iteratively on all the different cell types and see which cell type I should study in order to help the patients most. So just read that paper and answer those questions using either take a video or write an essay if you like, several paragraphs to explain those questions. Okay, that's your homework number 3, and then we will have one more homework before the semester ends. Okay. So for this, once you load the training data with responders, then you can start the session for the training. Your data may look different from what I have here. What I load is maybe different examples from this. So what I load is exactly this one here, okay? The data load in. But it doesn't matter as long as you have 2 classes. You have a training data, and then here I just go to the section of SVM. It's largely in this group. I can see like a linear separation. I don't need a tree. So this is the model I'm going to train. So once you select your algorithm, you can do training, and I don't need a parallel because there are so few points. Okay, it's done. There's one mistake. We know there's one mistake about this assignment. And that's a summary. This scatter plot is here. Confusion matrix, I don't need to plot the RC curves. Yeah. So this model is done, and result table. Okay, it's finished. So what you can do if you, in the real application, you can see, save this model after your training. You save this as a function. And then once you have new data, you just call that function and put the new data in. You will get the response. Okay? Any questions for this part? We finished the SVM. Easy. Now we can close this. Clean my data. I want to come to this page and then double click on this visualization projection. Found a way to select all the code here. Right click and copy it. So I basically copy all the source code from this M file. Close the rest. So come back to this clean command line window and paste all this to the command line. It should run this piece of code. This piece of code will bring up an empty window like this, and you are ready to use your mouse. Right click to create points. So I show you how to do it before you do. So use your click and start to click points. Just randomly make a circle. And once you get enough right points, then you by click once, right another button of the mouse. So if you click in the left and now you click right. So now in the middle, we start to click again to create some blue points. The code is inside the channel, and you click this to copy. Okay, I give you. So this basically show you a kernel transformation. You have right points around this, and then in the middle you have this different class of the points in the middle. So there's no way you can use a straight line to separate them in this 2D dimension. But this show you the transformation. It will make it 3D and make these points in the higher once you finish this Gaussian kernel transformation. Right click, it's hard to operate. So you just click and to draw this right points, then right click to stop. And then you left click to start to point the blue points, then right click again. So basically left click to make points and stop using right click. And then start to left click to add more points in the middle, and left click. That's to stop. That's all. This is a program that set it up. So once I finish my left click of the blue points, then right click to finish, then this program will transform this into 3D. And then you see the transformation allow you to separate the points afterwards. So, good. You don't have that. Oh, go up. No, it's inside this kernel. Yeah, that's right. Yeah, copy this. Yeah, you should be able to select or just copy Control A. Yeah. Yeah, you can start to click. Yeah, you're done?
Yeah,
No.
kind of like the door a little bit.
Okay, good. Yeah, just like that. Not yet. Yeah, you change the color by, you finish the right points, right? By you click this. Okay, now start to click blue ones in the middle. Yeah. Yeah. Then once you finish, then you click right button to stop. That's all. Okay. Yeah. Nothing happened. You should have that in. Oh. No, you haven't copied the whole thing. Did you copy the channel?
Yeah.
No, you need to copy all. Then this should work. Should have this. Yeah. It's hard to use. Right, left click. And then click left again. Oh, it's already finished, so you have to start another session. Close it, yeah. Just paste all country. You have to copy that again. Now we are going to reinstall the toolbox, single cell gene expression toolbox. So I encourage you to use add-ons. So now I'm going to delete the manual installed the folder. So I will do select this and just delete it. Because the new version, I allow people to install through these add-ons. and easy to update the version later. So what I can do is to I just select them all and delete. I have some trouble to delete files from this folder.
I could continue and it worked.
Really? Oh, that's good. So I want to have a clean installation, so that's why. Okay. Now I have no. Now if you do, you try to do this too, you shouldn't have. Okay. So that means we have a clean, clean uninstallation. So ready? Okay. So now I want you to use the toolbox as add-ons. So you click this. And from here to search. And then just install it from here. Oh. Yeah.
I'm trying to delete my file, but it's asking me for administrator permission.
Oh, really?
I can't find the compare admin rights widget on this computer.
So there's no way you can install this as a.
I guess not. I can't, unless I can find the software that would compare admin rights on this computer. I can't at the moment.
Okay, let's see. If you cannot, then you have to use the traditional way, just the unzipped. So I show you the. Yeah. So then you see. Do you have any this problem? You cannot install. You're still waiting. So I'm okay on my machine. I think on my machine this has been already installed as a add-on. Already, yeah, okay, yeah. So some of this machine we do. Where this is?
Yeah, the same problem as him. Because when I try to
Oh, so
remove this,
oh,
it
yes,
was asking
wait.
for administrative permission.
What I tried. It's gone. So from home, then add-ons. Have you tried? No, you're still not delete. Still waiting for, yeah. What I tried is this here. Maybe you need, can I shut down this? Yeah, some files locked. It's okay, but you can still install that. Can you do it? No.
Because it will ask me for administration.
Yeah, so you probably just need to restart restart the MATLAB maybe. Just some file, maybe source code that's locked because we are running it. But after you restart the machine, either the toolbox or start the MATLAB, you should be able to. Yeah. Even though you may not have the clean deletion, you should still be able to install the add-on. So you still try to delete the file. Yeah, I know, yes, but it's okay if you cannot delete it.
Okay, so
Clear, yeah, just keep it. Yeah, just keep
so
adding the add-on.
I run a transport. It'll
It still pop up.
yeah, still pops up. Can I try installing it in a different folder?
Can you just try shut this down, just restart, and then it's like a clean, so all the. No, don't run it. Yeah, because if you run it and it's locked up, just directly right click and delete it. Then you just use add-on to install that. Yeah. Okay, so yeah, so what I found is we can delete it once you restart MATLAB. Yeah. Because some of the function is running to log it up. Yeah, if you can. Please just wait. Maybe this functions in progress. Maybe just let it wait a little bit.
Still asking for admin permission.
At the end, but I think that most of the files have been deleted. Yeah, I had the same problem.
I'll just try adding one.
Yeah. So after the installation, if I do which, I should be able to find, okay, the toolbox has been installed in a local folder inside this add-ons, okay, toolbox. So that's where the new location as an add-on package is located. And we can use the tool as a before. So if you already have this prepared running on your machine, you can try to download this example data using accession number here. Okay, so you can visit this page and see what this is, and then come back to copy the ID and come back to here to import as a geo accession number. Oh, this is a problem now. Same error, yeah, sorry. Let's see if I can. Array. Yeah, so this is what you can do on fly here in order to allow this demonstration or exercise to continue. So if you found the error message here, so I will update later so to fix this bug. So what you can do is to click this error message once you receive it. Click this line 341. And you will be pointed to this line here. The difference you will have is this curly bracket around this default answer here. So if you add these two around it, then the program should work. Because the bug I forgot to put this curly bracket around this default answer. So if you add this two, if you add this in, the program should be able to run. Oh, you got a different answer. Okay.
Yeah, I have line 389.
389.
But it's different because when I go to 341, it's not the same as 3.
So are you sure? If you look at the place, where the same place?
It says error in SCG2 dash IN underscore document
the same file, but maybe looks like you have a different version. So you see here, this is the latest version we just installed. You should have that in there.
yes, the one that is
The same.
here. It's in here. That's in. So it's like a different.
They shouldn't be there.
Because it's in add-ons, toolboxes.
Oh, right here. See. So let's see what you have.
Security 9. So
Error using this, but it is still inside here. The same problem.
oh. So it's like when it calls this one. Oh, okay. This one, it's calling.
Yeah. So that's what I'm going to do. Highlight this. Found the line, which line? 340. Different. Yes, it's weird. I don't know. It's different file. So we download the same. Let me think. Yeah, it's weird. I don't know. Yeah, you already have that. Different. But I just download the same. Are we using the same version?
Yeah, I went to add-ons.
Why we download the same different files? Let me see. Do you have a version number? That's old. That's old one. I believe you are not using the latest version. If you do check which, yeah. So you are still using the old version. Yeah. So you didn't delete the old version. That's a problem. Yeah. So. So I come back to the old version. Let's try to find the old version to come back. So that's your older version. You need to delete it. I don't know why old version doesn't work. Yeah, we need to find a way to delete all the files. Yeah, we need to delete all the older files. Maybe just rename it. No, I don't know, because you still point to the older version, so you use. Oh, you need that to delete, which you shouldn't. Yeah, you have a different problem as we have, as I have. So you need to find a way to delete older version, then run from there. Oh, there's another way. Let me show you. What you can do is to delete the parse here. Not point to.
So from here it was giving me that option.
Yeah. So let's remove. Where is your?
Maybe from here?
No, there. Maybe there's a.
because in here it was giving me the option to delete that.
Uh-huh.
So
Yeah, yeah, maybe, yeah, that's right.
and then
How to do this?
indicate files not open. No. Okay.
No, I don't know. You should remove the path somehow. So I don't know if you have the latest version. If you can help me to fix this bug yourself, you should be able to run. Looks like some already have that in there. Okay. So, okay. So this should work. Then you use this accession number, download quite a lot of data. This is from mouse skin. So we get this, right? Everybody have this. Not yet. So once you have this, you click this button to do this one-click annotation. Select mouse as a species. Then just wait. And it's a good time for take a break and come back and to get the results. Yeah, right. Yeah, definitely need the help. Yes, I know. Okay, so if you haven't run this, don't start it. You just look, because this will take several minutes to finish. But at the end, you will get the annotation looks like this. Okay, so this is the same as what I presented in the lecture. We are going to look at two genes expression. These two genes are this ligand receptor pairs. So you can copy these two genes name, and then from here we can select genes, the first button here, and then you just paste these two genes name in there. This should be case insensitive. We should be able to find these two genes. We don't do normalization. We just look at the raw expression. So very hard to look at. Yeah, so this is basically the pair of the ligand and receptor. Should I use? I think the B, the subunit B is better. So if I replace it with a B instead of A, maybe signal is stronger. But anyway, so this is what we'll get. The donor cell, which is this group, contribute more about this ligand, and the receiver cells, which is this group, have a high expression of this subunit of the same class. So this is a stellate cell. Okay, so this is the donor cell for this ligand, and this is receiver cell for this receptor. In this 2D view, it's very difficult to see because the stellate cell has been embedded among this cluster of cells. So maybe we need 2D embedding in order to show. But anyway, so this is the idea about to visualize these pairs of cell-cell communication between different cell types. Now I think I can use this example to show you how to annotate the cell type using customized marker gene database. So those fibroblasts, endothelium cell, macrophage, T cells, they all are from the built-in database from BanglaDB. You can annotate the cell type using your own marker genes definition. Okay, so if you click this button here, for this window, for now we just say no. We're going to have this. Okay, so this is a small example marker gene database you only have two cell type 1, cell type 2. Cell type 1 contains gene highly express these genes, and cell type 2 has these genes. This is just a toy example. It's not real. It's just give you some idea what you should put in there. You need to have a cell type name, then marker genes around based on your own study. So once you have this database in, you can click, then allow the program to process. And then based on the marker genes, and one of the group has been annotated as a cell type 1. Okay? The others, there's no options, so it's still unknown. You can try again, because this time we'll generate a different random set of genes to see whether this is not real. So the randomly selected gene pairs maybe not give you any information about the cell type. So there's a lot of unknowns. But let's just use a real one. If I load another cell marker gene database information from SC type, then I load, this is skin data, so let's see which one I can use. Well, this database has no skin as a tissue, so maybe I just randomly select none of them. But I just select, for example, like this. So this is provided by SC type database. So if I use this as an annotation database, I should be able to get some of this annotation for those. So one of this is annotated as a megakaryocytes. Okay, so really based on your own applications, you can customize this annotation results. All right, in the last 30 minutes, we will see whether we can successfully install Python along this. Will be very difficult because it's a hybrid of two languages, two systems. But some of the analysis, for example, in this toolbox, if you look at the external, this function with the snake, which is Python, require the Python environment to set up. If for example, if you want to use this detection of doublets, okay, if you want to scrapulate method, you have to have a Python installed. After we installed, we need to visit this link to set up variant Python environment. So this interface knows which Python to call to communicate with. So now if you click this, the program will ask you, there's nothing happened, ask for a link which to point. So this Python environment hasn't been set up. We need to find in Windows, we need to find an executable file called python.exe. So you need to click yes to search for where the Python is installed until you found it. But we haven't installed the Python, so we don't have that. So once you found the Python exe file, then make a link, then these two languages can communicate after that. So now we need to install Python on machine. Follow the procedure I just developed for you to do this. Okay. There are several steps. First, let's check the comparability between different languages. So if you click this link in the item number A, visit this page. And to find your MATLAB version and also which one we should use. What I have is this one, 2023B, so I can install maybe 11. Based on that page, I cannot open it right now. So the highest number I can use is 3.11 version of Python. So 2001B, so you need to have 3.9, okay? So for this latest version, you can have 3.12. So I have this version on my machine, so I'm going to install 3.11, and you have to find your version, okay? So once we finish the decision of this, we come back. Number 2, so I view we can skip, we can directly jump to number 3, link C. So here you found which different Python version carried with a program called minicodon package around the Python to provide this interface. You can download Python itself, but there are other associated packages which help Python to run. It's better to run from here. So what we need, if you download the latest version, okay, that will be this. This is the latest version. But if I need 3.11, I have to find so this is 3.12. So I need to find the PY 3.11. So that's why the machine we are using is Windows. So we need an extension with the name with the Windows x86 and also PY11. So that's what I will download. So this file is what I am going to use for my machine. And if you use 3.9, you need to download 3.9 and that one here. Okay. All right. Of course, for Mac machine, you have to decide which Mac version you have. So I'm going to download this with 91.4. So this download. Then I can open the file to install minicodon, basically. And this is just for me. remember the location, remember where you installed your package, because later we will ask MATLAB to search for this folder to find the Python executable file. So remember, this is a C user, my name, then app data, local, minicodon. So I will work around and make sure you have the right. Yeah, good. So you know where you installed.
Yeah,
Okay, your same path. Yes, just remember, you have to point to it. You don't have that. If you follow the link here, it's number C. Yeah, I just found which version you want. Got it? Okay. It may take some time, yeah. So you download 1120, same one here, yeah. Mm-hmm. You don't have to, yeah, you can just jump to the C item in the list. Yeah, just found it here directly for yourself. The old,
Is it the same or?
no, this is the latest version, so.
Mine works with the latest version.
Okay, then you're done. Okay, yeah. But are you installed this? Oh, this is a PC, right? Yeah, yeah. I thought you were using Mac. Oh, PC, okay, good, easy. PC is easy. Okay, so just complete, just next, and then we are done. So now the Python should be installed on your machine. Let's just finish the final setup by itself. Now to use it, if you start your applications, you should see this recently added anacodon prompt. If you click, you can either click this black one should be okay, and then you should have this page, which is the command line window to communicate with anacodon. So from here, we are going to first install a tool called Git. So if you visit here, so now we already downloaded and installed this. Now you copy this codon install. Okay, install a small program called Git. Git is to help you to download other packages. Okay, it's just this. So once you finish run this, then you run the next, which is the whole line of this. This allows you to download and install single cell 24 XCT into the Python environment. Then we are ready, we are done with the installation. Yeah, you're already done. So now you can copy the next, this line from, yeah, the whole line, yeah. Then just put it in there. And just wait, then you are done. Okay. So have you finished? Yeah, so you're running the installation. That's right, yeah. So those are the very typical operations in using Python. So you install Python package using this way. Say yes, yeah, just click. It's good. Still running, yeah. Take some time, the large package. Okay, yeah. So it installed a lot of dependencies. Error message for the second. The first one is done, right? Yeah. Yeah, let me see. No, you don't have a Git. Once you finish this, you just install. Yeah. Because the Git this rely on the Git. Yeah. Yeah. So once you have the Git installed, then you just come back to this page. Copy this. Okay. To run this and then install. That's all. So you can find this information in the tool, this XCT2, the website. Those just procedures. Once you are done, come back to this page. Okay. Now we are set in the MATLAB, the Python side. Come back here and by click this to set up a Python running environment. You click this and then see locate Python. So now we need to find inside. So this folder could be invisible because this is like a systems folder. The Windows doesn't want you to see. So you have to maybe type that in mini codon until you reach to this page, this link. Then under this folder, you should be able to find python.exe. So I'm in this folder now, and then point to this. I'll give you time. So if you don't know where it is, look at this, the folder directory, and this inside here. So once you select this file and click open, then the location folder will be saved into MATLAB, and then the link between two languages has been established. Is that all? Okay. So I keep on this page, just make sure you know where to find the parse. So you only need to do this once. Once you're saved, next time you still have this established. Unless you uninstalled Python and change the folder and replace by something else, you may have two different versions of Python installed in parallel in the same machine. So in that case, you may want to switch. Then you just found a different version of the Python through the change of the folder name. Found it? No, it's not.
I thought it's the same path.
No, if you do, I show you. If you do a data and then local, then mini is inside here. Then you see. Go down. Yeah. Mm-hmm. Then open. Then you are ready. Yeah. It's okay.
It'll be fine for the Python thing.
We are going to use smaller example to do this.
Okay.
Yeah, we are not going to use it's too big. Yeah. Okay, so once you click this saved, then it should be okay. You don't have to. So we are not going to use the data set with a large amount of the skin cells. We are going to use a small example to run this because the data is too big for this 20 minutes. Okay, so now we just set up this. Okay, yeah, we're ready. Yeah, we are not going to use this example. You said, okay, good. Yeah, question. You don't know where to find the Python, no problem. Yeah, so this is done. That's good. No, it's here. Where's your name? Where's your name? There. You don't see this, so you have to type that in app data data. That is /local. Then / yeah. / then mini m. So inside here. That's it. Then you just open it. Then you are done. You should be able to. Yeah, set up. So you don't need it to find here. Okay. Yeah, good. So I think all of us have already set it up. So we are going to use a small examples to make a XCT run. So so you still have error message. You still don't have a Git, so that's a problem. I don't know why you. Yeah, so you need to, there's something wrong with the git problem. Yeah, so you have to do that. So now we are on the same page. I'm going to use small example to do this. So we do import data and to load the example data, okay, rather than load a processed example data. We just discard this huge data set. Okay, so this one is a mouse data and contains few genes, few cells. Still, if you look at the genes, there's some genes. It's already very clean gene, but let's make it smaller. So you can basically run on this, but for the purpose of demonstration, I will use network and also remove some genes to reduce the search space to make this quick enough. So select the mouse, and this remove genes, including remove some less interesting genes, remove non-coding RNA genes. And also, the most important, you can set the last into 2000, so you only keep 2000 highly variable genes at the end. Okay, now I have this smaller data set. So I'm going to use this to run in network, then one sample cell-cell communication. So you click network and one sample. And then there are introduction page for this method. Continue. Basically, it's saying your computer has not enough memory as recommended. What you can do is either you just ignore and try and see whether you get luck to run, or you can just prepare the everything essential as input and the source code, and you remove the whole thing to a better machine, okay, in Python. But for our case, we can just proceed anyways, and then you select a folder to save your results. So I'm going to save the results onto my desktop here, and you can select other folders you can remember. So the results will be saved, and also the Python we are using is this, okay? So this is what we set for the Python environment, if you can see on the screen. So this is correct, and I can say use this. Now it's time to select the cell type. Remember, the communication is between two cell types, so I need to select two cell types. So I'm going to select endothelium cell and alpha cell, so two cell types. If you select three, it's too complicated. Then the program will say, no, I only need two. So you have to remove one. So every time you have to pick two cell types. But next time, if you want to study endothelium cell with beta cell, you just select them, run that again, okay? So now I have two cell types. Then this is directional. So I want to use endocrine cells as a donor and alpha cell as a receptor, or other way around, they're different. You can run both, okay? You will get a report from both sides. But I see, in this case, I select one, and then the program just run. You just wait until the final results will be pop up, and in the folder, you decide where to save. Okay, so internally start to build a network for one cell type, build another network for another cell type, build corresponding networks in between. So you see a lot of this intermediate steps. And it's also called this parallel process to use all the cores on your machine. So you see some of these pop-up messages. All you have to do is just wait until the results come out, okay? So the building of network is native MATLAB code. Then there's one step using the Python part. We have not translated into MATLAB language, so that's why it depends on the Python, one of the steps. So I will leave the program here and just like to finish off today's session. And let me know if you have any questions. This may finish, may not, until 12 o'clock. Okay, it should take 5 minutes or so. Depends on how fast your machine is. The result will be in the folder you decide where to put, and it's a text-based file, CVS file, so you can open them in Excel spreadsheet, and you will see columns of the ranked link between ligand receptor pairs. Okay. Let me know if you have any questions, okay?